Start.
Hello, good evening everyone. Myself Tejas and I am going to explain the topic about immune response against virus and fungi. Next slide, sir. So there are about millions of species of fungus, but among them only 400 to 500 are potential fungus, which can cause human fungal diseases. So as you can, these species are how our body bounds the immune response against them. So next. So obviously fungus can enter our body via several routes such as entry by inhalation. So the fungal spores can enter our body via our nostrils. It can also entry through injury sites. Let's say there is some of fungus spore and there is injury in our in your hand. It can easily get entry through your circulation, and lastly it can other mucosa circulation, and lastly it can be get associated with your gut mucosa or other mucosa lining, and they are like a kind of opportunistic pathogens. So whenever you put opportunities pathogens, when they you would get opportunities, they would get in. So there are multiple modes of entry, right? Next, sir. Now, fungal diseases or mucosa are classified based on few following criteria. So, first of all, site of infection, where it is superficial cutaneous, subcutaneous, or deep and systematic. So, these are important factors that you think about. Then, the route of acquisition, it could be either endogenous or exogenous, coming throughout inhalation, or might be present already in gut mucosa, our gut line, and lastly virulence, whether it is primary or opportunities. So against these fungal pathogens of our body mount two types of immune response. Next slide. In innate immune response, which is highly specific. So that innate immune response components involves the macrophages, dendritic cells, neutrophils, and complement, where they are adaptive immune components such as the T cells. So next slide. T cells create cell of populations. Relevant cell wall components of the fungus includes glucans, polymers of glucans, mannose, long chains, glycogen polymers of N-acetylons. All of these components can work like a PAMP or gens. Associated with molecular pattern that PAMP recognize the pattern receptors in the dendritic cells or macrophages. So there are many pattern recognition receptors such as TLR249 that can determine the components, whereas lectin, C-type lectin receptors crucial for recognition fungal pathogens. Associated molecular vectors with these receptors of innate immune system are able to understand that indeed there are in fungal infections more into fight back and against that. So the fungal species would be optimized complement proteins. So here we can see complement proteins can coated by candidate albicans. Now when these complement proteins are decorated the outer surface, macrophages can really come and bind to these receptors. These complement proteins are macrophage of component receptors, and the end of the results in engulfance of these fungus with the help of macrophages. Other than that, there are lectins which can bind polysaccharide type of antigens, and it would lead to the fixation of components, and the components mediated damage ultimately kill the fungus. Fungus are also intelligent. They have many strategies which prevent the immune system to harm them, which one of the fungus that creates some kind of like capsule around them. As the result, the dendritic cells is unable to recognize it at the first step case, or the complementary proteins are to settle down at the surface, and these fungus is actually survive and can infect the human, and this kind of fungus is actually dangerous. Next slide. Now, now it has been known as the AIDS patients suffer increased incidence of mucous candidiasis and many other fungal infections that as HIV, when the helper cells are suffer from number of cell are now they are susceptible in fungal infection, that in turn means the fungal towards the infection involves the intermediate responses. Next. And these days it is well known that Th2 cells, ones of populations of cells are really important against fungal immune system. Th1 of the cell populations is macrophages and creates inflammation. These to more macrophage recruit and effluents of the fungi. There are the clearing of these fungi from the system. Now, next one. Now the recently of Th17, IL-17, which is the key factor effect against the fungal infections. Next slide. The Th17 cells are generated when the nice T cells are activated in the presence of IL-6 or TGF beta, but the question is where does the fungus cytokines come from? And the answer is these cytokines is treated by dendritic cells and macrophages. Once these dendritic cells or macrophage recognize fungal pathogens with the help of TLR2, in this case, this signaling would lead to the IL-6 or TGF beta production. As a result, the T helper cells would be differently with Th17 population, which plays an important role in the fungal immunity. Now we can understand that the how innate and adaptive immune holding the each other hands compared with the fungal infections. Next. In summary, we would look at what it is the fungus of immune response against the fungal infections. What are all the recognitions methods of the body sector of cells infect the fungal infections? And finally, the mechanism of fungal defense response, how innate immune system and adaptive immune system will coordinate each other to prevent the fungal infection that we had go throughout these slides. Yes. Immunity against viruses, some of these viruses like TLR3, TLR9. In these, we can understand which DNA in form of viral origin. So they are trained to recognize viral TSDNA or TRDNA, and these how the macrophages or the dendritic cells understand this DNA from virus and viral attack. Now, once they understand there is a viral attack, what measures do they take? It's like some of these T helper cells will in turn activate T cells and make plasma cells. With these plasma cells will secrete the hell out of the antibody against specific viral proteins. Next. And the virus would optimized by the antibodies. Now virus need to interact with the host cell and membrane receptor within surface glycoproteins, and in order to facilitate the insulation of virus inside the host cell. But if the virus already optimized by the antibodies, the interaction between the host cell membrane and the viral glycoprotein would not locker the results. It's hard for the virus to get in. Not only these, many of these macrophages and dendritic cell is expressive with the receptor, which can bind to the F region of the receptor and intake the viruses for bacteria and break them apart and the digestive system of the lysosomes. Next. Now, other than the macrophages and dendritic cells, the other cells such as CD8 cells and killer T cells play an important role against the viral protection. So the CD8 positive cells have specific CD8 core receptor, and they recognize antigens on class 1 MHC. Let's say the virus can infected by the epithelial cells moratile in yellow. Now the virus has entered the cell. It would uses its host protein synthesis machineries to create viral proteins. Next. Out of some of these viral proteins will be mistake degraded by the protozoans inside the host cell, and these degraded protein fragments would consume the endoplasmic reticulum via the tap transporter. Next. And load to the class 1 MHC molecules. Now Gans and platforms of the name suggest the platforms would create a pore to which the granzymes is the not got in host cells. Now in the host cell it can activate the big or bags, which are for provence uptake genes, which it would ultimately lead to the cleavage of inactive captors, and it would allow the inactive captors to get activated, and as a result a of this would occur the CA positive T cells express Cas8 ligand, which would interact with and also can activate Cas8, and thereby it can include Cas. Next. The end result, the death of the target cell along with the virus, which was growing inside. Now you think killing the cell, how does it help? How now killing these cells, two important things happen. Firstly, the viruses are inanimate stuff when outside of host. So first of all, their habitat is destroyed. Where they are growing happily is destroyed. Second, the dying cells, before it dies, it sends off an alarming signal, and these alarming signal are receptor by the neighboring receptor signals. These alarming signals, known as interferons, as a name suggests, it would interfere with the viral processes. Look, that matters is detail. Next. So the interference created from the infected and dying cells would bring to the receptor to the nearby infected cells, and there is an interferon signaling cells which translate into the uninfected cell. Ultimately, it would get an antiviral strategies which would be used by the infected cell, that if these unaffected cells is infected by the viruses, they can easily counterattack the jack. So the interferon, now you have other strategies. For example, you have next 2,5 oligoasynthesis, which would a sort of create particular protein which would bind to the viral tsRNA, and it would be inhibit and inactive RNAs L family molecule with jack of viral genome. Now then there are other helpful substances which would block the inhibition of viral assembly, and these protein complexes are known as mixed protein. So this is how an interferon based system interferes with the viral defense mechanism. Next. Though the interferon signaling works by the Jack carries several Jack phosphorus nucleus, but nucleus gives rise to special antiviral proteins, and depending upon the which stat combinations work is products is also different. Thank you.